Good evening to all friends and viewers who've joined this webinar in India. Good morning to those who are joining us from the West, Europe and the US. A little over a year ago, when the first lockdowns were announced, the IIT Alumni Center, Bengaluru, started to host webinars. And in fact, the first two webinars that we had was on the COVID-19 issue that we were all facing. The first webinar was with Satyajit Mayor, who you will hear today, along with Dr. Shahid Jamil of the Wellcome Trust. And the second webinar, if I recall right, was with Dr. Dhananjaya Dendukuri, who's on the panel today as well. And along the way, we also had an interesting webinar with Dr. Rama Govindrajan, who talked about fluid mechanics and how that works in the context of the virus itself. This was what we would probably today call the first wave. And by the way, all these webinars are available on YouTube on the IIT Alumni Center Bengaluru channel on YouTube. Now, pandemics can be scary, but as you will see today, we need not fear it. There is science, and if you follow science, I think we don't need to fear it. Looking at data on the web, you know that between the years 1831 to 37, an influenza pandemic swept across Europe and had three waves in 1831, 1833, and 1837. So there was three waves. Similarly, the Russian flu of 1889 killed nearly 360,000 people in Russia alone. And of course, the well-known 1918 Spanish flu that we nominally say began in 1918, but some researchers are claiming it started a little before that. But that killed an estimated 50 million people in three waves. COVID-19 has entered its third calendar year. If you take the first detections in November, December of 2019, we are now in 2021. And we've lost almost 2.8 million people globally to this virus. So we need to understand what these waves mean and therefore understand it and figure out how to combat it and remove the fear. The reason we are doing this is that there's so much going on in social media, in media, that's selectively chosen without the proper context and creates a lot of confusion. So today we've got eminent scientists to join us. I will not go through their bios today, but Satyajit Mayor is the director of the National Center for Biological Sciences in Bengaluru. Professor Gautam Menon was with the Institute of Mathematical Sciences and now at the Ashoka University. And Dr. Dhananjaya Dendukuri is from Akira Labs. He's a founder of Akira Labs. These are three people. We've curated a list of about 35 questions broken into sections, and they will answer all of them. For our audience, please put all of your questions in the Q&A box only, not in the chat box. We don't monitor the chat for questions, we just monitor the Q&A box. So between my colleague, Dr. Sushila Venkatraman and myself, we will monitor this and ask the team here to respond. So again, to re-emphasize, the objective of this conversation is to understand issues on a science basis. We will not take anything that is non-science. We don't want to get into those areas. We want to remove fear from the minds of people because with the appropriate behavior, I think we can fight this pandemic effectively. As the late Professor Hans Rosling, the well-known TED superstar, if you will, I think he had once said, there is no room for facts when our minds are occupied by fear. So let's get that fear out so that the facts can come in, and let's get this conversation going. So I will ask the first set of questions to Professor Mayor, if I may. The first ones, here are the first questions for you, Satyajit. What do we know about the COVID-19 virus today versus a year ago? And how do we know this? You're on mute, Jitu.
Yeah, thanks, Ashok. For having me back, and two, just to reiterate that I think as you said, if we know more about the virus and its operations, there's sort of less to fear because we then know how to deal with it. And I should say that from a year ago to now, there has been an extraordinary explosion of our understanding of how this virus. I mean, a year ago at this time, when we had this, when we had the talk, we knew the size of the virus, we knew its its genome to some extent, which had already been sequenced. We knew a little bit about how it gets across from person to person. We knew something about the virus as it infects a cell and the consequences that it has inside a cell. Today, our understanding has really exploded in the sense that we know precisely the receptors, not the receptor alone, but the receptors that the virus now takes to get inside cells. I mean, of course, we knew that it was tropic for a certain type of a human cell which carried the human ACE2 receptor, the angiotensin receptor, but the angiotensin, exactly, the angiotensin converting enzyme. But in fact, there are many other molecules at the cell surface, as we know now, that can attract the virus and recruit the virus to its, in a sense, into the belly of the cell. We know from work from many other groups of what the consequences of the virus engaging with these receptors. The virus seems to have a program that allows the cells, allows it to get inside the cell and in fact recruit other cells because it changes the surface of the cell it interacts with and creates in some ways an attractor that now makes other cells fuse with it and produce a sort of a sequestered environment in which it can grow and multiply. I mean, and what I should add is all these bits and pieces of knowledge that we have also come up, have also allowed us to target the processes that allow the virus to infect and replicate inside a cell. I mean, it is still early days, but we do have many molecules that are in very late stages of clinical trial that are now combating these very fundamental mechanisms of how of what we have learned over the past year. So so the knowledge, I should say, from the virology point of view, from the point of view of the virus and the cell, the cell it inhabits is monumental. Yeah. So I will stop there. Yeah.
So this, I think the second question, therefore, is then how does this virus spread? In fact, one of the viewers here in the Q&A box says, if I keep my windows open, will the virus float in?
Right. So I think that's a superb question because the virus.
Let me just finish this.
Yeah.
I think the Lancet in April 2-3 weeks ago, had published an article that said that this was airborne.
Right.
And if you recall, a year ago, we were all taking these Savlon sprays and spraying it on the surfaces of whatever you had.
Right.
There was even an ad on TV where the guy was spraying the book that he was reading. So, but now you're talking about airborne. So if this is airborne, then how does the population protect itself from the spread?
Right. Well, and the two parts to this. I mean, and I think you actually had a beautiful seminar from Rama, where she was talking about the generation of these very fine particles, right, aerosols, and that is in fact what happens. The virus is present in aerosols that we generate. These aerosols can be large and small. The large droplets that are created when we spit or speak or eat are in fact do carry the virus with it. But when we breathe, we also breathe out, we also generate aerosols, in which the virus particles often are encapsulated. But these aerosols, are of a size range. I mean, some of them are very large and they drop down and fall on surfaces. And it turns out that what falls on surfaces doesn't seem to have a very long lifetime, unless it falls on a surface where the aqueous environment of the virus is preserved. But these aerosols that are generated are sort of, encased in sort of, almost a monomolecular layer of water, which allows the virus to be in some ways active as it floats about, like dust particles. And we know how dust particles move about in very, very small dust particles move about in the air. So if you keep your window open, just to answer the question from the person in the audience, these aerosols, if somebody has, come by and really sneezed very loudly near the window, or very harsh, very vigorously near your window, these aerosols are likely to, come into your window. But remember that the aerosols are also being generated when you speak or when you sneeze, and they're being dispersed in the air. And a very, very small fraction of that contains the virus. So the likelihood of your little nostril of that certain cross-section, picking up that virus is very, 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 very small. I mean, in fact, it is infinitesimally small. But if you're in a room where many people are sneezing, talking, engaging in conversation, the air is laden with these aerosols. And now the chances of your picking up something in that tiny nostril of yours is very, very high. So, I mean, it's literally a question of concentration of these amounts. So the generation of clusters of people in any location is actually of a very, of really explosive nature. And that's really where the virus it can be enhanced in its transmission. And it's turning out that the viruses that we have today are more transmissive because perhaps there is more of them in these aerosols rather than on these particles, on these large droplets that go flat. But again, the jury's out on the distribution of the virus particles in these two types of particles that are generated by us. The prevention is obviously masks, and masks that can actually are not just, unfortunately, not just cloth masks, because cloth masks do have large pores. One does need double, triple layered masks that can actually prevent these small particles from percolating through the material that you breathe, and percolating in both ways. If you're infected, if you have a mask of that nature, you will prevent the spread, and if you have a mask, you can also prevent the ingestion. So both ways, a mask is really the best vaccine of all. Yeah.
Okay. All right, so that really leads to the question of the various mutations that we are now talking about, right? So what are the variants of this virus? Are they stronger, more harmful? Just if you can educate all of us about where it started, where we are today. There is this B117, many of these variants going around. They call it a double mutant. There's all kinds of names.
Can
you tell us what is what? And
sure.
Even if you have to use the slide, it will be fine.
Okay. Okay. Thanks, Ashok. I'll address that. I'm going to sort of first make a small disclaimer. I personally have not been studying the variants, although we have been studying the virus and how it enters cells and all that, which is why I know a little bit about that. But I have, in anticipation of this variant issue, because it has become extremely, important for us to understand, I have stolen a bunch of slides from colleagues of mine who have been working with this. So let me first share my screen, and then have you sort of just go through some of the slides that I have prepared for this. Yeah. So here we go. Right. Can you see my screen?
Yeah, that's fine.
Okay, so just to very quickly, give you a rundown of what the virus is. The virus is this particle here. It has got a lipid bilayer with little molecules that are called the spike proteins that are poking out. It's about 100 nanometers in diameter, and therefore very happily incorporated into these aerosols. Now, this virus has a large numbers of, well, it has 30,000 nucleotide sequences, 30 kilobases, and it has about 10 to 14 open reading frames, and it makes about 24 to 27 proteins when it infects a cell. Of course, when it doesn't infect a cell, it is a dead particle. So it is in fact completely useless. And a little bit of soap on the membrane of this virus will completely destroy it. Or if you heat it for 80 degrees, because it's so fragile, it's finished. But in the aerosol, it is actually retained for a while. And this virus, then, if one actually then follows its gene sequence, and I'm going to now make a plug for what Chitra has given me today about SARS-CoV-2 variants. And Chitra is, in fact, a former student of the NCBS, but now is an assistant professor at NIMHANS, where she actually studies a variety of different viruses. And she has done extensive work on sequencing of viruses in Bangalore, and along with other colleagues of hers, like Ravi, the person who we call Virus Ravi, and Anita, Dr. Anita Desai. So there's their work, and I'm going to just simply breeze through some of the slides that she gave me. So again, as I mentioned, the virus itself has got this sequence of 30,000 nucleotides. And if you look at this at the same sequence that I plot, or she has plotted below, looking at about 4,000 virus genomes from December of last year to April today, just looking at 4,000 such virus genomes, what you see is these little spikes. And these spikes literally reflect total number of amino acid substitutions found in the gene sequence of this virus from the beginning of the time that we started tracking it. What you see are these long spikes. I hope you can see that on your screens. And these long spikes are where there are many, many, many mutations. And there are literally some gaps. And these gaps are places where mutations are disallowed. In fact, if there were mutations there, the viruses probably never survived. In fact, so the variants, if you will, are literally, if you can think about the permutations of these various little spikes that you have here, every little permutation of that spike is a variant. Okay, so I mean, as you can see, given this number, there seems to be a variant at almost every location inside the sequence of this virus. I mean, figuratively. So there are very, very large numbers of variants, but not all of them show up. So, so what, in fact, my colleagues have done is they have asked, how do you track these viruses, and what's the lineage that one may actually have in, oops, sorry about this. These things are coming in the way. Yeah. So starting here, which is at the beginning of when we began to see the virus, there was the A lineage, and then nested in the A lineage, derived from that, accumulating certain sets of mutations, we began to see the production of the B lineage. Then, given the mutations in the B lineage, we derived the B.1 and the B.1.1, and these are simply nested sequences derived from the original sequence of the virus that was first, for example, sequenced in Wuhan. So these sets of lineages are therefore the different variants that are circulating today. And they reflect the sort of and following the the collection of mutations across this whole sequence, you can actually position them within this sort of a tree that gives you a lineage map of what obtained from where. And often these lineage maps are and these origins of each of these mutations are associated with single epidemiological events. So you can track them from these, literally geographically and temporally track these lineages in space and time. So today, these lineages, there are about globally about 800 lineages floating about. And if you look at the, and there are some variants that actually become variants of concern, which is really what the question you asked. And those variants of concern are variants that begin to have mutations that confer on the virus properties of enhanced transmission or enhanced infectivity, or maybe enhanced pathology. So far, we don't really know of variants that can give you enhanced pathology, simply because the epidemiology of that is not yet complete and not well understood. So the variants of concern are variants that, of course, can give you enhanced transmissibility or enhanced infectivity, meaning things that can be transmitted better or things that can infect better. And those, of course, are going to propagate in a much larger way. So, I mean, for example, the variant that right now was the one of concern in early November was the variant derived from the lineage B.1, which is B.1.177, which is the variant that showed up in the UK. And sorry, B.1, and the variants that came out from B.1.7 that came out in UK, which carried mutations that really made it hugely more transmissible. And very quickly, this variant began to overtake the different variants that were present in the world. So Ashok, just interrupt me if you think I'm going to.
I think yeah. So I think the key one right now is the P1. Is it the P1 is the one that's scaring a lot of people?
P1.1.7 was the one that showed up from the UK strain, which began, and I have a time map of that here in India. And in fact, this is the time map from the UK, where it showed up in September of last year in the UK here, and then it spread very quickly across the globe and began to dominate the population of global lineages. Mutations on top of that have also emerged, and these have emerged independently as across the world, and mutations have emerged also in India, and in Maharashtra as well. So I'll just bring you to something that we see in Karnataka right now. So for example, in Karnataka today, I mean, this is up to April 7th, so it's about a month old. We have sequenced at our center about 700 sequences, and about 65% of them up till April 7th were very generic, spread across that entire set of lineages. But there is one, which is this B1.617, which is the so-called double mutant that everybody talks about, that was first picked up in Maharashtra, also showing up at very high frequencies already by April 7th in Karnataka.
Yeah.
And here are the kind of charts that one can generate if you do serious genome sequencing of these variants. For example, today you can see, look at this bar here. Not today, 1 month ago. If you look at this bar, we have about 80 sequences in this week, in the week of the 7th. You have about, maybe 50% of them coming from this variant B1.617. Whereas, maybe a few weeks before that, very little of that variant was present. Now, this is, important because these variants, if they spread more rapidly, more people can get infected quickly. And therefore, you can enhance the R0 number that I'm sure you're going to get to in a little bit in a while. So I'm sort of got a few more slides on the variants, but I'm going to hold them because I think we should get on to some other questions as well. But I hope I was clear about what variants are and what their origins are. And I should say that any virus that is made produces variants because their intrinsic mechanism of replication is very sloppy. And in fact, that's also an Achilles heel of the virus. If we can enhance its ability to make much more mutations, the virus will also fizzle away because most of the mutations it makes are crap and it cannot circulate.
So what you're saying is that the best vaccine is masking and potentially using a double mask, as they say now.
Triple, possibly. Yeah, triple layered mask, I would say. Yeah.
So that's probably the best vaccine that you could have
today.
And, be a little more careful when you're in the company of, so if you're walking on the street, stay away from people. If you're in an apartment complex, don't go into the lift with 10 other people, that kind of stuff, right?
Yeah.
So I think it's time to get Sushila to ask questions on some definitions and stuff like that. Would be useful in taking this discussion to its next level of infections. So Sushila, can you start?
Yes, thanks. Gautam, maybe we'll start with you. And what we need help with is, we come across so much of terminology in the literature that we come across, forwards that come to us or stuff that we see in the press. And things like fatality rates, positivity rates, recovery rates. There are so many of these terms, and very often it's all very confusing for us because we don't really understand exactly what they mean and why should it be important to us. So if you could talk a little bit about that, would be nice.
Sure, happy to do that. So let me start with the first one, which is R0, or the basic reproductive ratio. In a sense, that's the most fundamental way of thinking about an infectious disease. It's a one number that encapsulates all that is really important at an epidemiological level about the disease. So what R0 says is, what is the number of people that one person who is infected in a background of many people, all of whom are susceptible to the disease, will infect? Do they infect 2 people on average? Do they infect 5 people on average? Do they infect 18 people on average? That's what this R0 measures. It's usually defined right at the point where an epidemic takes off, because that's when people are not yet prepared. They haven't changed their behavior to avoid people who they suspect may be ill, someone who's coughing, etc. It's just life as usual. And it's in that background that you really want to ask, what is this fundamental property of the disease? So some numbers, for measles, that number is very large. That number is 18. So one person with measles in an unsuspecting crowd of people who haven't had it before will give that infection to 18 other people. For Ebola, that number is 2. It's somewhat harder for Ebola to move between people because you need direct bodily contact with some bodily fluid of someone with Ebola. Most diseases, influenza is probably somewhere around 2. And there is a debate, of course, about COVID-19, but the number is probably somewhere between 2 and 4, the best that we know. But remember that this is that basic reproductive ratio. That's something that we don't see anymore because that's right in the beginning before we knew more about it. Now what newspapers and people write about is something called the effective reproductive ratio. That basically says how much is it going to be amplified between a day or 2 days or 3 days, etc. So it's much more of a local measure that already accounts for the fact that we're prepared, we're masking, we're avoiding people, we're avoiding crowds, etc. So that's what the reproductive ratio is, and it's very useful because if those of you who remember this old story of the king and the wazir who the wazir is asked, what do you want? And he tells the king, I just want 1 grain of rice on the first square of the chessboard, 2 on the next, 4 on the third, 8 on the fourth. And it just, he says, just give me whatever rice there is at the end of it. And you would think, looking at the numbers 1, 2, 4, 8, 16, that it's never going to get very large. But on the other hand, by the time you get to the 64th square, that's a huge, huge, huge number. It would have bankrupted the king and the kingdom and the country and probably large parts of the globe several times over. So that's what's funny about exponential growth. It really gets to very, very large numbers very, very fast. And that's really what the reproductive ratio is important at conveying. That fact that you can amplify that number starting from just one person who can infect other people. So fatality rates, so a bunch of different definitions here. One is called the case fatality rate. That is, for people diagnosed with the disease, what fraction will die? So suppose since the beginning of the COVID-19 epidemic, we have had, let's say, 200,000 people die. But a number that is 10 times that has been recorded as people who've been infected. I'm just making up these numbers. They don't apply to COVID-19. I'm just giving you an example. That fraction of 10% would be your case fatality ratio. That's to be distinguished from something called the infection fatality ratio. The infection fatality ratio is people who've had the infection, but who may not count as cases because they've not been tested, they've not shown symptoms. And the funny thing about COVID-19 is that these are very distinct from each other. There are lots and lots of people, this whole background of people who are asymptomatic for the disease. That is, they will show no symptoms. For every one person who's tested, there are probably 20 or 30 who are not tested and yet have the disease. So the case fatality ratio is easy to measure. You just count the number of people you've tested, you count the fraction of them who've died, divide one by the other. The infection fatality ratio is the far more tricky thing, and you really have to infer that from the data, from the sorts of tests that you're doing. It's much more indirect, but in a sense, it's much more basic to the disease itself. The case fatality ratio depends upon how well you identify cases in the first place. So that's the property of the medical system. But the property of the disease is the infection fatality ratio. Recovery rates, how many people have recovered, divided by how many people you've registered as cases right from the beginning. And then let me see what else. Test positivity. That's how many people have been tested over a short time interval and how many of those tests turned out positive. Right now, India is at levels of about 20% of test positivity, which means roughly 1 in 5 tests is turning up to be positive. That number should be as low as possible to indicate that you have some control over the disease. The WHO recommends 5% as your cutoff. 20% is way above that. In some states, we're seeing 30%, 35%. So that's roughly what you should look at. It doesn't make, often many of these numbers are computed over the last 24 hours or the last 48 hours. There's a lot of noise and jitter in that data. It's better to look at it averaged over about a week or 10 days or something. Then it's smoother. Then you ignore the local ups and downs in daily testing, and you get a number that you can trust more. So that's test positivity. Recovery, how many people have recovered divided by how many cases you've had, typically since the beginning. And this is, of all of the numbers that you can think of, this is the most useless, because you know that most people will recover from the disease. Right in the first week or 10 days, you have lots of cases and no deaths at all. It's only a little later that people will begin to die. And by the time you reach right at the end of the epidemic, you know that COVID-19 actually doesn't kill too many people. It kills, the case fatality is probably somewhere around 2%. The infection fatality rate is probably around 0.1%. So right at the end, once everything is over, you might expect that 2% of the people who you registered as cases will actually die. So that would be the right, and the rest of them will recover, which is 98%. So you hope to get to that 98%, starting from zero. There's only usually one way that curve can go up, which is up, barring little wiggles when you suddenly have a rise in cases. So that's, of all the numbers, as I said, that's the most useless. That's the one, unfortunately, that is used very prominently in the news. Yeah. So that's all the numbers. I hope that helped.
Thank you. So, you know, the other question is about this first wave and second wave. In some ways, like the first, we don't seem to have come out of the first wave at all. So what is all of this about? Is there some sense in this first wave, second wave? Is there definition? Is there something that you see the end of the first wave and then start a second? What does all of this mean?
Okay, so that's the terminology of a wave actually comes from long back when people were thinking really about things like measles, which were very seasonal. They sort of went up during school season, then they plateaued to zero, then they went up again in a very, very periodic way. So roughly speaking, between the two waves, you saw nothing at all. That hasn't been the case here. I mean, you've had a the first wave peak at about 98,000 cases. Then you came down, but you never dropped really below 10,000. So it sort of stayed there, and then it sort of took off in the second wave. So people use the terminology waves because you see an up and a down and an up and a down. It's not clear that this is applicable because people have been infected throughout. There never was a situation where infections went off to zero and then suddenly reappeared from somewhere else. So I think when people talk about waves, they refer to this up and down. There seems to be a difference qualitatively between the first wave and the second wave, in that the second wave seems to be driven more by variants of the type that Jitu was talking about, that these variants tend to be more infectious, they tend to move more rapidly between people, and also behavior has changed, which contributes to that. People sort of took it a little more easy starting about mid-December, January of this year. So waves is not completely correct. It's an informal way of talking about this up and down. Something has changed between the first and the second wave. So in that sense, in that very limited sense, it's okay to think of this as a second wave caused by something new that wasn't true earlier. Very roughly, that's the story.
Okay. And now we hear that there is a third wave that is going to come in, and we're going to see more younger folks getting infected. at least anecdotally, we are seeing much younger people now getting affected, losing their lives and so on. So again, for example, I'm part of a Rotary COVID war room where we get these requests for beds and oxygen and all the rest of it. And we are seeing that there are people 35 or 40. And then yesterday we got a kid who was 10 years old also being affected by this disease. So is that a sort of the start of a third wave, or a change in the way the second wave or whatever is happening is going on? What really is happening there?
So there are actually two questions in there, and one is a question of multiple waves, third wave, fourth wave, whatever, can they happen? The answer is you're not guaranteed to have it. There's nothing that says, even though the PSA was quoted as 2 days ago saying that there will be a third wave, you have to prepare for it. It all depends upon what one does now, what people do now. The transmission of the disease is a very social phenomenon. It depends upon how people get together, how much, what precautions they take, how much mask wearing they do, how much of the fraction of the population you can vaccinate. So it's completely possible that you can actually hold out against a third wave. There is no guarantee that there will be a third wave. That's one thing. So that's not ruling it out, but I'm saying that there is no crystal ball that says you have to have a third wave by August or July or September or something. That's really not necessary. If you can manage to be cautious enough, there need not be a third wave at all. That's one point. The second point relates to the age. Usually in the first wave, across multiple countries, it's been more elderly who've been affected in the first wave. And by the time you get to the second wave, it's younger people. And this is believed to be roughly something to do with behavior and behavioral changes, maybe not more than that. Right now, we are seeing an overall lifting tide of a huge number of cases. You're seeing it in Bangalore, where you've already crossed the limits on your ICU capacity. You're seeing it in Delhi, all of the major cities. So you're seeing younger people also because you're seeing more people in the first place. And even if the young were 10 or 20% of that, you're seeing that enhanced by a factor of about 10 compared to what it was in the first wave or earlier. So if you were to look at it statistically, I mean, there's a difference between the anecdotal information that you or I receive about our friends, about people we know. On a statistical basis, as yet, there is no very strong indication that young people are overwhelmingly being affected in this wave. Still, the risks are still carried by the older people, not by the younger people. There may be a little shift in the sense that people who caught it the first time, older people, also are now more older people are vaccinated currently. And you can see that very clearly in the data for the older people, that there is a definite improvement in the factors or in the fraction of older people who would otherwise have been on this line. Now they're distinctly at lower risk of being diseased with COVID-19. So right now, anecdotal about young people. It's possible that you may be seeing it. We may actually see this in the data in the coming weeks. But right now, there's nothing really to suggest that this is not just a fact that there are more people altogether who are being infected.
Just one other definition, some question which has come in the Q&A box about what is disease transmissibility of a disease? How would you define that?
So I think of it somewhat informally in the sense that, it's like the reproductive ratio. One person who's infected is sort of maybe instead of infecting 2 people, may infect 3 people or 4 people. That means that the disease is more transmissible between people. That roughly seems to be what is happening between the original strain and the new variants that you're seeing. The new variants just are a little more efficient at moving between people because they tend to bind better, to attach better. The virus attaches better to cells, to the proteins that Jitu mentioned on the surface of the cell. So that's what we understand. That's the sense in which I would think of it.
Okay. Maybe Ashok, you can take over and let's talk about testing.
Yeah. So ever since now, more than a year, we've been. I think the general thing is we've got to test. The only solutions are testing, testing, and testing. So we bring in Dhananjaya, who's has done a tremendous amount of work in this area. So Dhananjaya, can you start by explaining the importance of testing to the layperson? Why should we be testing so much? When should people test? Should people in certain jobs test more? So the guy from Swiggy or Zomato or whatever the courier services are when they come delivering, should we be worried about that? Should they be testing more? Security guards in offices and so on. So can you just talk about the importance of testing briefly, and then we'll go into the specifics of different tests.
Sure. Thanks again for having me. I think the major sort of learning from the past year has been that more frequent testing is actually a good thing, even if it is less sensitive, right? So there's sort of an overemphasis maybe on PCR testing, and indeed PCR is maybe the reference standard in terms of viral RNA testing. But I think there is a growing school of thought that cheaper but less sensitive tests like rapid tests are actually as important, especially as we're talking about opening up the economy. And to your specific question about what sorts of people should be getting tested, I think, anyone who is going to work, going to school, right? I think testing these sorts of people as frequently as possible is only a good thing and will really help open up the economy. Having said that, I think the RT-PCR test will continue to be the reference standard in terms of its sensitivity, and especially in any cases related to hospitalization, symptoms, and so on. I think you do want a confirmatory RT-PCR. The other kind of, I think, high-level learning is that a positive is a positive. So if you have a positive, whether it's a rapid test or an RT-PCR, it's a positive, right? There is no need to probably second guess and double check that. However, a negative test is slightly more complex to interpret. A negative on an RT-PCR is also very likely negative, but a negative on a rapid for a person that's also showing symptoms at the same time should be treated differently, and those people should likely go get tested again, ideally with an RT-PCR. That's, I think, some of the high-level things on testing. So I would say overall, more testing, more frequent testing, even if it's not as sensitive, is the way forward in terms of COVID testing and opening up the economy.
Yeah, so let's go a little deeper, in the sense of how good are these tests when you are conducting them at such large scales? How good are they? Number one. Are there, since the outbreak, we would have learned a lot. Have there been improvements in the test processes, procedures, and so on? That's a second question. And the differences between antigen tests and molecular tests. If we can just kind of talk about that.
Sure. So just in terms of definitions, I think everybody already knows this right now, but the two major tests for actually detecting the virus are the molecular tests and the antigen tests. The antibody tests cannot actually detect the viral presence in the body, so they cannot be used for this purpose. So the molecular tests and the rapid antigen tests are really the two major categories of tests that are available in terms of in vitro diagnostics. And if you look at the way both these tests are done, they have very different principles. The molecular test is actually looking at the RNA of the virus and exponentially amplifies it using PCR. So you have billions-fold amplification, which is why it's extremely sensitive and can pick up down to a copy of the virus in a microliter. That's a very, very small amount of virus, right? Whereas the antigen tests are not picking up the RNA material of the virus, they're actually picking up viral proteins, most likely the nucleocapsid protein, which is in abundance in the virus. So that is picked up using a format that's quite similar to the home pregnancy test. And these tests are quite easy to do. They don't need instruments. They can be interpreted even by lay people with a little bit of training. And as we all have seen so many pictures, they can be done at railway stations, malls, really at point of care, even at home. Although you're not allowed to do that in the Indian context, right? So the PCR obviously needs a lab and a little bit more infrastructure.
Yeah, so one of the questions is, with all these variants, does the PCR capture all of them?
Yeah, that's a sort of a kind of nuanced question, actually. So the PCR is very much dependent on the particular PCR test and the primer design for that particular PCR test. And as Jeetu talked about, you have all of these mutations along the way, right? So if it so happens that the primer set that you have used is actually not designed for a particular variant, you could miss that. But that miss is actually interesting in a way that you could use PCR and some clever combinatorial PCRs to effectively pick up these variants, right? So the reference standard way of picking up variants, I would say, is next-gen sequencing. And for surveillance, that's what's being done. But that's not possible to do at a very large clinical scale. But if you actually design your PCR tests appropriately, and you know that there's a limited set of variants floating around the population, you could use sort of a combination of PCR tests to, at least with a high probability, identify which variant it could be. And in fact, that's one of the products that we are looking at putting into the market soon with one of our partners.
Okay. As you may imagine, this is a very popular topic. So our Q&A box is actually flooded, and we're trying to weave in as many questions into our questions as possible. So Dhananjay, one interesting question is, do we ever get to see a home-based COVID kit? You said pregnancy kit. So do we have a home-based COVID kit?
So the answer to that is, in the US, there are already home tests approved for home use and interpretation. In India, the guidance right now from the ICMR is that testing is to be done by labs and professionals. So this is not available today for home use in India. All testing in India has to be done by labs and personnel that know how to deal with the biomedical waste, deal with samples, deal with tests, and so on. So technically, it is possible such tests are available, but they are not legally available to Indian home users at this point.
Okay. I would like to throw googly at you on mass spectrometry. You can back off and say you don't know the answer, which is fine. But would mass spectrometry or any other similar method be a better way of testing for COVID than RT-PCR, given the concerns about false negatives?
First of all, I think false negatives with RT-PCR is, I would say, I know there's been a lot of stories in the media about people only being picked up in CTs and not in RT-PCR and so on. And we haven't actually got to the bottom of that. What's happened also, I have to say, in the Indian market is that the reference standard way of doing PCR is ideally a 3-gene test, right? So when you do 3 genes of the virus, you're very likely to at least pick up 2 of them, for instance, even if there's a mutation. However, there are many 2-gene tests now available on the market. In fact, many tests are being done in this way. And it could then happen that one of the genes doesn't have a proper primer design. The other thing is not picked up for some other artifact reason in the lab, and then suddenly you have a negative, right? So we don't exactly know why this is happening. It could also be improper sample collection and so on. So I think it is not necessarily to be blamed on the RT-PCR is, I guess, what I want to say. In terms of the mass spec itself, I'm not an expert on this, but from what I understand, the mass spec really picks up potentially bits of the viral protein. And I think to do today, what's happening, especially with our kind of case loads, is we need batching and we need high throughput. So to set up mass spec to do very high throughput testing in a lab is still, I think that's not an easy problem to solve. So yes, it can be done conceptually, and I think there are enough research papers showing that. But if you're talking about, I mean, there are labs doing 10,000 samples a day. So these people, in fact, do not want to hear about any innovations and new things. They just want to crank out their results on things that are working. So that is the situation of the labs today. And so while it could be done, they're unlikely to adopt it at this point.
Yeah. So your opinion on a couple of things, Dhananjay. So many variants out there. What are the implications for diagnostic tests and the future of disease surveillance? Number one.
Yeah.
Second is testing capacity in the country and availability of tests. Do you have any comments on that?
Sure. So starting with the rapid antigen tests, I should first of all say that a good thing in some sense about rapid antigen tests is even if you accumulate a few mutations, it's unlikely that you will lose the sensitivity on the rapid antigen test. Okay. So even with whatever variants there are, there's probably no reason why you shouldn't be picking them up with the rapid antigen tests at a reduced sensitivity. With the PCR, like I already mentioned, depending upon the specific primer design, you might miss that. But on the other hand, one way of looking at it is that you could cleverly redesign those tests to pick up variants. And that's something that could be done. So I think given how easy it is to redesign the PCR test and actually adapt it to these variants, you could do that. In fact, we have had situations where customers from Delhi call up suddenly saying, you know, your RDRP gene is not lighting up, but the other 2 are, right? And it comes consistently from certain labs in certain areas in Delhi. So you know that there is a certain mutation that is happening there, right? So that's actually interesting epidemiological information for us to be gathering with the PCR and should actually be contributing to surveillance efforts. So I don't see a reason why you can't pick up variants, I guess, with the PCR. And in regards to the capacity question, I just have a slide, if I could share it for a second,
sure.
Which is So just so that this is just based on public information actually. So the total approved labs today in the country is about 2,500 for COVID testing specifically, correct? And we have tested over 30 crore samples at this point. So in some sense that you could say that's almost a fifth of our population, although it's probably a lot of repeat testing. And on the most recent day, we tested about 18 lakh tests, correct? This on the right side is a table that I just calculated, which is labs per million people based on ICMR data and just population in India. So these are for all the major states, correct? And a few things I guess stand out. I mean, if you look at, again, the last 5 or 6 rows, in some sense, you could predict that these are the states that will likely have very low labs to sample, labs to people ratio. And as this wave starts spreading from the west of India to the east, I guess that's one significant concern is that several of these states that are actually kind of to the east or towards the east have very poor lab capacities. And that could significantly diminish our ability to identify people with COVID. And although people are in real time augmenting lab capacity, especially in the private space, because of the demand for testing, there's still going to be significant challenges. On the other hand, I should also mention that a state like Delhi, which has actually a very good, in some sense, by Indian standards, lab to people ratio, still there are significant challenges and delays and so on in terms of getting test results. And obviously this is to do with the density of the population as well. So I think we have a long way to go in terms of this getting the right test capacity. You also have a lot of practical issues on the ground where most labs that we know have infected personnel. So they are not able to turn around tests in time because some people just don't show up for the shift. So there is a lot of challenges right now in terms of lab capacity. And I think in that sense, the rapid antigen, again, is something that we should be looking at more actively because it might just be our only option for the next few months to augment. The other thing I'd like to mention is in terms of just rapid antigen versus PCR, this is an interesting publication from last year done in the US. And I want to mention this because this has some relevance to India as well. So the point that they're making in this plot is that this is a typical viral load. It goes up very sharply and then it falls in a typical person. This is the days since exposure. So it starts rising after the second day, goes up and then falls. And this is obviously an average plot. It could have variations for individuals and so on. Now a PCR test, a typical PCR test, would detect as low as this. So it would identify somebody quite early on, okay? And a typical rapid, not a typical rapid, but the rapid that they've chosen in the US would identify about 2 logs off, right? Which seems like a big number, but because the virus grows so fast, it's just about a day. That means on day 2, for instance, the rapid might miss it, but on day 3, it would pick it up, right? So if you frequently test, you could actually pick it up even with the rapid test. However, my own knowledge of rapid tests in India is that we do not have rapid tests that are as sensitive as the ones that are reported in these publications. My guess is that rapid tests available in India are more likely around here, which means you probably have about a 2-day differential between a PCR and a rapid test turning positive. So these are some of the things to keep in mind. Having said that, it will still pick it up eventually if you do it frequently enough.
Okay. Thanks, Dhananjay. Let's go back to Jitu. Everyone's talking about genome sequencing, okay? And so one is, what is it all about? And if genome sequencing is important, are we doing sufficient levels? I think your slide also showed a very small number, but as of 23rd April, I think in India, they showed that we did only 15,133 samples, which is less than 1%. As opposed to 4 to 8% that's been done in the US and UK. So what do these numbers really mean? And Jitu, go off, you're on mute. Oh, we lost him?
Yeah, sorry, yeah. Okay, first of all, genome sequencing in general, I mean, it's the most sort of common genome sequencing that we all know about, and the traditional one is Sanger sequencing, where it's very, very slow, laborious. Each virus is sequenced individually. Then we have something called nanopore sequencing, a slightly higher throughput, where you can do on a single machine maybe 12 samples at a given time. And then we have this next gen sequencing efforts, which is basically what allows us to sequence at scale, where in a given run, in these large machines, you can do several hundred viral genomes at the same time. And these sequencing efforts typically take about a day to conduct the sequencing. I mean, and as I said, depending on the technology, you can do several hundred genome, well, 1 to several, or even 1,000 genomes in a day. Analysis of that data, so to ensure that you got the information correctly, requires another few days. Right. So by the time you've got the sample in your lab and you've got the sequencing done, it's about between 3 to 4 days, and then you're able to say you got a virus from an individual, and you can sequence the variants that call out the variants that individual viral genome has. So that's sort of the sequencing technology today. And what we have seen in India is that there were rather independent efforts right from the time the virus entered our shores. In fact, Chitra, the person whose slides I showed you, she has been sequencing the virus right from the time that the virus showed up on our shores, and so have the IGIB in Delhi and CCMB in Hyderabad. They've been sequencing these genomes, on a small scale. Today, our capacity in the country is on the order of about 2,500 to 3,000 sequences a day, which is again really small. But now we are all connected in the form of a consortium where the NCBS, in collaboration with our campus neighbor InSTEM, we've created a laboratory like 10 other laboratories have across the country to use all the resources that we have of next-gen sequencing to actually sequence several hundred genomes a day, and that is something which we are going to need if you're going to track the presence of these viruses. A coordinated effort across the system, and the Indian SARS-CoV-2 genome sequencing effort called INSACOG is the effort that has now been put together. I mean, it has been, I should say, slow in taking off, but it is now, I guess, running as fast as it can with the installed capacity that we have. I mean, the capacity means these large sequencing machines that require feeding of both money and technicians, which again is of short supply in the country. So, but I think being able to do strategic sampling across the country, and we do it frequently enough, is a task that the INSACOG has taken on. And I can see that now we've also got a set of new members as part of this consortium. Maybe 15 labs now are doing it, and soon this will be an effort that I think will have decent coverage, right? So in terms of sequencing, I mean, this is where we are at. Ashok, I forgot the second part of the question. If you can just quickly repeat that.
So the first part was to explain genome sequencing, which you've just done.
Right.
And the second part was, are we doing enough?
Right. Well, we are not. Yeah. So we're not doing enough because it's really, each sequencing effort is several thousand rupees, anywhere from 3 to 10,000 rupees, depending on the sequencer you use. So it's enormous cost. It also requires a huge amount of infrastructure and this analysis pipeline at the back end. I mean, everything is possible, but we didn't have the resources in the beginning, or they were not thrown at us to generate this. Although some of us were saying this should happen, this didn't happen fast enough. But now that we have some momentum going, I think the idea would be to feed that momentum and ensure that we build up the sequencing to at least between 5 and 7 to 10% of the sequences that are present, so that we get a good coverage and are able to foresee the mutations that may begin to take over long before they do so. Right? I think that's the way we can stay ahead of the game. And at the same time, one needs to have laboratories that are able to take these sequenced genomes and ask these sort of questions about viral transmissibility and its capacity to be more infectious or less, and most importantly, evasion of vaccines, right? And that's something that also needs to be augmented in the country. Right now, very few places can do that, and this is again something that we have been sort of really insisting on from the scientific community anyway.
Thanks. Sushila, you want to talk about vaccines?
Yep, I think this is a good point of time. Jitu, there are many questions that have come up where people are asking about wanting to know a whole lot of questions about the vaccine. So maybe if you could spend a little time, tell us what types of vaccines, the 1 dose versus the 2 doses,
right.
When should I take it? Once I get vaccinated, why is it that after the 2nd dose, so many people are falling sick? So all of those, please.
Right. Okay. So let me say there are, today there's an explosion of the number of types of vaccines that are now available for SARS-CoV-2. More vaccines for this one infectious disease than the combined armory of all the vaccines that we've had, for every disease so far. I mean, it is just, remarkable that within a year, one has maybe 6 different types of vaccines for a single infection, for a single infectious agent. And these are, let me just run through some of them. These are the, beginning with the sort of the vaccines that are somewhat at the moment out of reach here, which are these RNA vaccines. The RNA vaccines are literally packaged RNA that encode information about the virus, which can be delivered into our cells and then our cells, and they then, translate the information about the viral protein. This vaccine is, is really the front end of the technology right now, because it can also be produced extremely quickly. The time to make an RNA vaccine from the obtaining the sequence to actually having it distributed is 3 months. I mean, this is just unprecedented, and it's simply because this technology has been developed over the past 10 years for actually 3 different types of diseases, and the NIH invested a huge amount of money in the Moderna vaccine is precisely an outcome of that, which in some ways is more distributable because it can be transported, again, due to just some simple inventions of transportability of this extremely fragile molecule, the RNA. And again, innovations that came from recognizing there's going to be a need for this. And then, of course, the Pfizer vaccine, which still needs very, very low temperatures to be transported, but that's, again, the mRNA type. So the mRNA type, as I said, is the cutting edge of technology right now for the vaccines. But we also have, the old technology, which is the inactivated virus, and several vaccines, in fact, including the biotech Covaxin, which was, where they've taken the ICMR strain to make an inactivated virus. That's another, that's perhaps the oldest technology on the block. And that's also available, but that takes some time to build up stocks, because it does take time to produce the virus and make sure that it's properly inactivated, go through all the QCs, and then that virus. That vaccine does take a minimum of 6 months to build and develop a decent amount of stocks. Then there's the stuff in between, which are the vaccines that are, for example, the protein subunit vaccines, which there are many different types. Some of them are already in use. I think there are from Russia and from China. There are the vaccines that are coming out are of some protein subunit vaccines are coming out even from the US. There is then the and these vaccines again are proteins that are targeting the spike glycoprotein of the virus, so that when you introduce it into the the animal, that's us, you generate antibodies towards the spike glycoprotein, and that gives you protection. The vaccine that is of a slightly different kind that has come about again in the past 20 years, I'd say, are these adenovirus vaccines, which is what Covishield and the AstraZeneca vaccine are about, and the Johnson & Johnson vaccine that is in the US, where and I think Sputnik is also an adenovirus vaccine, and from Russia. So these are these are vaccines where people are using a virus backbone, which encodes also a gene for the corona virus gene, again the spike glycoprotein, and that adenovirus is an adenovirus from, in the case of the AstraZeneca, it's from a really rare adenovirus from a chimpanzee, and in the case from J&J, it's also some even rarer adenovirus that's not circulating in humans. So these adenovirus vaccines are in some ways DNA-based vaccines, which again, when injected, encode for the viral protein, and they generate the protein inside our system, and then we mount an immune response against it. They're again also rather relatively easy to make, and the technology for this has been hugely, I should say, enhanced. Now, so this is the spectrum that we have today, and I must say the extraordinary thing is that almost all of, well, I shouldn't say almost all, I think all of them have significant efficacy. It really is important that that the vaccines themselves, one should remember, have been put through even more paces than any other vaccines in the history of vaccination in the world, well, not just for the period of time, but also for the large numbers of people that were recruited to doing these tests. And I think in some ways, here in India, we didn't have as much transparency as we should have on some of the efficacies of the vaccines that were introduced first. And that created a bit of hesitancy amongst the population in using the vaccines that were locally generated. But one could argue that this was an emergency, but one could also argue that had we waited a little bit and got the efficacy data from the Covaxin, people may have been more willing to take on the vaccination in a more full-bodied manner. Now, the reason for the 2 doses is actually interesting, because in many cases, in fact, for some vaccines, you don't need 2 doses. And I think that depends on the quality of the immune response that the human species mounts against something that's injected into it. And in fact, most of the vaccines, as we note, are 2-dose, except for the J&J. And they are because that when you get the first dose, you're tickling the immune system to generate antibodies. And the second dose really boosts that sufficient to provide the protection that we believe is required. The J&J, for some reason, and I'm actually not a vaccinologist, so I cannot give you the precise reason why, the J&J seems to do that in 1 dose. But having a booster is not a bad idea, because what a booster does is actually take the very high affinity antibodies that are being generated and really enhance the production of those so that you have significant amount of neutralization capacity in your system to really neutralize any variant that comes along or any vaccine infection that comes along. Now, a last point which I know you asked was that about does one vaccine not provide, one dose does not provide protection. One dose does provide protection from severe disease, but it does not provide protection from infection. And I think we are seeing that as the variants broaden out and their range increases, we are probably going to see less and less capacity for protection against infection from most of these vaccines, and therefore they're going to have to be tweaked. They're going to have to be tweaked, and again, we know what the pipeline will be. The mRNA vaccines can be tweaked faster than the rest. Tweaked so that they really encapsulate the variants so that we can get protection against them as well. I'm going to stop there. But I think it's very important that one brings on people who have public health experience with vaccines, for these discussions about what happens in a population. It's very dangerous to let a scientist who's works in the lab let loose on these questions, except to give you reasoned answers.
Fair enough. I think we will look for someone to do a follow-up, webinar. Couple of other specific questions around immunity. Are we actually seeing it happen? Do we have data that shows that immunity? The other is specific to an individual. So after what is being told is that immediately after the vaccination, the immunity of a person comes down. Is there any truth in that?
Yeah, let me take the second question first. I think there's no, let's just dismiss that because there is really no truth in that. And I think it's dangerous for, people to be propagating such, such information because it's simply not correct. Going to a vaccination center, if you're not properly protected, may actually expose you, as we said, because aerosols are around, to infectious agents, but one has to be properly masked and therefore, make sure that you don't get infected there. But the fact that vaccination would give you reduced protection, I think that's really an unfortunate, canard that's being circulated. And I think one may sort of refer back to when people get vaccinated, you feel that in fact, I experienced that as well. When I got vaccinated, I felt that, I'm protected. And, your guard may come down, in terms of being, your own behavioral guard may come down. And that could contribute in some sense to one's, seeing some sort of enhancement in infection. But I'm not sure, I would certainly dismiss the argument that vaccination reduces your immunity. Now, whether the infection itself creates immunity, it certainly does. And one of the reasons why the spike in the first instance went down is because there was some amount of immunity that was built up in the population since large numbers got infected. But we don't know fully the time course of the buildup of that immunity. We don't know whether that immunity builds up in everybody or whether it builds up in a subset of people. And it's turning out, as more and more data comes in, that the immunity doesn't build up in everybody. Many people who are infected do build up immunity, but there is a small subset, and we don't understand why. They don't actually build up protective immunity. But maybe they don't build up protective immunity even when they get vaccinated. So these are questions that really need immediate attention from people who are looking at immune monitoring of what's happening in this disease right now. And we don't have predictors for that at all, except to actually do a test in your own blood and ask whether you have antibodies to COVID and whether those antibodies, even if they're there, whether they're protective or not in a neutralization assay. That can be done, but it's a real I mean, it would be impossible to do this for a population.
Thanks. Dhananjaya, maybe we'll come to you, and can you tell us about what happens with these vaccinated as these new variants come in? How does that work? And does something happen?
Yeah, again, I should preface this answer with the same disclaimer that I am certainly no vaccine expert, and this is based on my reading and understanding of the scientific literature. I think one misconception certainly is that if a vaccine has been designed with a specific antigen, right, then somehow it will not protect against other antigens. And I think that's simply not true and is borne out by lots of clinical data now, actually. And the reason that is that the body actually kind of splits up these proteins into small bits, right? And even though you might have a few mutations, there's large bits of it that your body probably still recognizes and is able to deal with it, right? So once you're vaccinated, your immune system can learn. It's just like human beings learning, right? Once we learn a certain formula, it's not like we only remember the answer to X question. We know a lot more than that. So I think, first of all, scientifically, there is no reason to believe that vaccines cannot take care of mutants. And what's even more gladdening now is that there is existing data that is available, both in vitro data that maybe Jitu can speak about, that shows that the Indian vaccines are actually able to neutralize the variants. And there's now data very recently from the last week in Qatar, which showed that I believe the Pfizer-BioNTech was very good, again, neutralizing against the South Africa variant, which was of concern, and actually prevented 97% of all severe hospitalizations across a fairly large set of about 30,000 people. So I think the vaccines are doing almost better than people expected. And so we should be very cautiously optimistic about this.
Great. I think we have Sushila, so I'll just take it forward as, and we are also coming to the last few minutes. This is probably the most challenging part of this conversation because it relates to numbers and its interpretation. Gautam, I'm going to put you in a spot, but we need honest scientific answers here because that's very, very crucial. Okay. So to roll out, I think our official tally of COVID-19 deaths so far is about 218,000 or so that they are talking about. But general opinion in society is that this is a gross underestimate. Okay, so first, what is your estimate of the truth of these numbers?
So we think that the undercounting is probably somewhere between a factor of 2 to 4. So take the number that you had and multiply it by some number between 2 and 4. The exact number you should multiply it with depends a little bit on which part of the country you're talking about. The cities do it a little better. It also depends upon which wave you're talking about. Now, all of these calculations are completely out of the roof because now there's real evidence of very, very serious undercounting, probably by a factor of 10. There's a genuine, huge discrepancy between government numbers for mortality and what people are just seeing with their own eyes. And brave journalists going out and counting numbers of bodies that are going into cremation centers and so on. Yeah, so right now the situation is very confused. Overall, probably 2 to 4 in the earlier wave. Now probably tending towards 5 or more would be the best guess.
Yeah, I think there was a TV thing with the Barkhadat, and I think Shahid was part of that, and he broke down on that interview. I mean, Gautam, again, this guy Jamie Malik, writing in the Hindustan Times on 27th of April, says the rolling weekly average of deaths has been increasing by 50% each week for the last 4 weeks. In fact, last week, he said it increased by 84%. So our fatality rate jumped from 0.7 to 1.7, from February to April. So does this suggest that we no longer take comfort from a low case fatality rate? Also, what do these numbers say about positivity rates?
Okay, so I mean, there are 2 parts again to that question. One is that this assumed exceptionalism of India in terms of deaths and fatalities was probably wrong. We were just not counting deaths correctly. We were not accounting for the fact that the Indian population is younger on average than the US or the UK population. So all of these make a huge difference. Your chances of dying are exponentially large the older you get. They sort of really increase very, very sharply between say 10 to 20 versus 60 to 70. You put all of these numbers in, put in the undercounting of deaths in, etc. You get numbers that are kind of comparable to what they have. I suspect that the current increase in case fatalities may be related to the fact that people are dying who should not be dying. It's just that they have not been able to access medical care in time, not able to access oxygen in time. These, given what we know about case fatalities, provided you don't overload the medical system, you expect a certain number based on your experience. We have now exceeded that number, suggesting that people are dying who need not have died. They're dying just because the medical system cannot keep up with those, with the current pressure.
Yeah. And, what do you think is the undetected increase in cases each day? I mean, if you switch on the TV, you will hear last 24 hours, we had 350,000 new cases and so on. I think you yourself had said that it's 20 to 25 times more. And the last serological survey suggested it was 27 and a half times more. The IHME, Seattle-based IHME, they said it was 29 times more. So what is the view of you guys on this? And since you guys are studying this very deeply, what does this mean for people? What should they do to stay away from infections?
So at least about the counting, I mean, our number for 20 to 25 was based on the previous wave. Okay. From what our analysis of roughly what should have given a certain level of mortality, given a certain number of observed cases, what should have been the underlying number of cases. I think anyway, I mean, it's sort of hard to estimate these things. You know, what is the severity of an infection? What do you mean by someone is infected if they can catch the disease again? What significance is that? And these go into questions of what immunity you're provided by a prior infection. I would be happy with a number anywhere between 20 to 28. I would think that's roughly the right ballpark. It's certainly not 10. It's probably not 50 or 40. Right now, the difficulty that I'm seeing is there are many people who would like to be tested but cannot be tested, especially in the cities and definitely far away. If you look at the reporting that is coming out from rural UP and other states, there are reports of whole villages where there are people with coughs and fevers, etc. But the nearest PCR testing site is 80 kilometers away. They don't want to go there, and they don't trust the RAT test, and this testing is not available. In the big cities, even in Delhi, if you want someone to come to your house and get yourself tested, that takes about 4 to 5 days, and it takes a further 2 days for the test result to come. So by then you're pretty much over the hill as far as your symptoms and your infectivity to other people are concerned. So now we're reaching a state with such high test positivities that people who ought to be tested under normal circumstances are not being tested. That widens the gap. People are not reporting for testing because they think that, look, I don't want to stand in a big line with lots of people who may be infected in order to be tested again. So I'm going to opt out of that. So there are many, many, many uncertainties in this, and these uncertainties are just getting bigger and bigger. This is really the problem with having a high test positivity, that all this casts a huge cloud on any sort of calculation or projection you can be doing. Unless you push that number down by testing more and by getting some sort of control over the cases themselves, you will be in a bad position as regards data. So right now, all calculations are out of the window.
So, if you recall, probably towards the end of 2020 in the US, when their numbers were peaking, they were doing what, 250,000 cases a day?
I think they never had a significant, they never had significant numbers. I think right now our numbers have outstripped everybody.
Yeah.
And our tests have collapsed.
And Professor Brahma Mukherjee's math projections are showing it going as high as upwards of 500,000 cases a day. I mean, is there a view on that?
So our own projection, I don't like to talk about these numbers and times and peaks because they're so uncertain. I mean, this is a question that I'm asked sort of a lot of the time by the press and other people. Everybody wants some sense of surety or certainty, which is very hard to give because the whole background of data is such a messy background. So our calculations done for a few weeks ago suggested that somewhere around the middle of May might be when you see some sort of relaxation. There is a very large bar on that, maybe 2 weeks before, 2 weeks after. So somewhere between the middle and the end of May. The problem is that you'll never really understand the scope of this because you're not testing enough and test positivities are so high. So the peak that you may be seeing may be an artificial peak because you've run out of testing capacity and you cannot test more. Whereas the true peak may lie later, but you will never see that as the true peak. So which peak are you talking about itself becomes a question. Right now, I think we're nearing the sort of peak as far as testing is concerned. So I would be very surprised if the actual numbers of cases went above, let's say 500,000. We had predicted 500 to 600,000, but as I said, that's just a number that's completely nominal. If you can't test for that number, you will not see that number. So hopefully things will improve, I hope, in another 2 weeks. That's the best that we can say. There's some slight hint that is hovering around, around 410,000 per day the last 2 to 3 days. Maybe that sort of says that we're saturating. I wouldn't bet on it at this point.
I guess like the audience, audience has got, there's some 50 odd questions that we still haven't been able to answer. We'll try and send those questions out to you. And if you guys do feel like responding, do let us know, and then we will share it with everybody. For all our viewers, we are going to, this is being taped, so you will have it up on YouTube on Monday, because clearly there's a lot of information that's being thrown at us. And you may want to look at it and understand it at a little more leisure. So Sheila, do you want to go through your last questions? Because somewhere they've answered pretty much all of what you had in mind for your last question.
I think there are 2 or 3 questions that we could kind of discuss just to wrap things up. And that is around what can we do about it? We've got, we've kind of talked a bit about it even as we were going along. But one of those is a question that came from one of our viewers. What kind of intervention strategies would help to reduce mortality? Is there something that can be done? Is there something that, as we become more aware about the disease and the science around it, perhaps there are steps that could be taken to have a better intervention at the day 5, day 6 kind of time frame, which is when the viral load is very high, etc. So is there something that can be done about it?
Maybe I can just add one thing there. I think the most important thing for reducing the mortality is very simple. It's basic medical amenities. And, it doesn't matter what else we do, that is going to be the one single thing that will reduce mortality. Increased oxygen supply and access to a set of protocols, medical protocols that allow people to overcome what today we don't really have a handle on actually the really severe ways of dealing with mortality from this virus. So people are still throwing the kitchen sink at the virus and the infected person who is very severely infected, and hoping that the individual is going to survive. There are hugely improved strategies, dexamethasone treatment, and certain drugs that are beginning to be repositioned for SARS-CoV-2. But the one single factor is really availability of oxygen. That's so basic. And, really, for me, I find that the most difficult thing to accept, that we have not planned for that. And,
so here's another question, and maybe I would request Dhananjaya to come in on this one, is that, someone has said that oxygenators are required more in the home than in the hospitals. What do you say about that?
Again, my views are based on talking to several doctors. So the oxygen concentrators, I'm assuming you're referring to those, they are useful in 2 circumstances. I think especially when people's SpO2 drops, but between about 90 and 94, right? At that level, these can produce enough oxygen to actually support those patients. But as it starts dropping even below, they are no more useful. So in that sense, it is useful, especially in contexts where you're not able to get a bed easily. And as you know, that's also happening in our cities, not to mention in the rural areas and so on. So there's definitely a big role for these oxygen concentrators early on in the disease, but one must remember that they cannot actually help you if your condition deteriorates below an SpO2 of about 88. The second use case for them is actually for people who have recovered, especially from very serious infections, who still need some oxygen support when they go back home. So even in that context, these oxygen concentrators are useful. So these are the 2 places that they find use.
Thanks. Jitu, one of the issues that we kind of briefly touched upon when we were discussing earlier was spillover. But I wanted to sort of contextualize it with what Gates said just after the Ebola outbreak, right? When he said that the world will struggle to deal with a pandemic, with an epidemic, which was what he had said. And, and we are seeing that happen now, world over. But what can we as individuals, as communities do to kind of help ourselves in a situation like this?
Yeah, no, I think that's a very profound question, Sushila, because it really goes to the heart of why we are in such a state, right? And I think there are many factors, and these relate to the huge sort of exploitation of the planet that we have engaged in.
Mm.
It also deals with one actually very singular point, and I should bring this up, and this is something that I also encountered in David Coleman's book called Spillover, where I mean the biomass that humans and human activity has captive is, as opposed to what is wild, is completely skewed. I mean, we and our sort of domesticated animals and plants sort of have now maybe about 80% of the biomass of the planet, and of course, where else do infections go, right? Where do viruses go? The number of viruses, the number of infectious agents haven't changed. And since we have encroached so much, right, on the planet's biomass, there is no other place to go. And 7 billion people are a fantastic incubator, right? Well fed and completely now in some ways the perfect test tube for generating all kinds of infections. So, I mean, so where we need to go from here is really begin to understand how we got here and whether we can begin to, at least jig some of these relationships that we have with our own ecosystem. I mean, to me, I don't think there is a solution that is short term. We are probably going to see many more epidemics or pandemics of this kind when the virus, begins to acquire these characteristics. So we should be aware and be vigilant of what happens in the future. But if you want to prevent it from happening and creating such an impact, we really need to think about our own species' exploitation of the planet.
One last thing, and this came from one of our audience who asked this question. Is there a lot of research going on in India? And clearly from what you've been saying, yes, the answer is yes. But we don't seem to hear about it. So what is happening, and how does the lay person really understand what is going on? I think it'll give everyone a sense of hope and a sense of comfort that, things are moving in some direction.
Right. No, I mean, there is research going on, and being a scientist, I would say there must be 10x or 100x more for a country of this size for us to really make a big impact. But I think the most amazing thing is the fact that research is not a local endeavor anymore. It is a globally connected endeavor. And as long as we have scientists in our country willing to engage globally and engage with, just the enormous amount of possibilities for collaboration, connections, and creating sort of knowledge and know-how for these challenging times, we are in a good place. And I must say that this has been the evidence for the past over the past year that the scientists, not just in India, but globally, have been engaged. So whether you're working in Africa or you're working in United States or in the UK, you are connecting yourselves to, we are connecting ourselves, globally. And I think that, to me, that brings hope, and that we are part, just as much as the virus is spread because we are a global village. Science is also now part of this global village, and is working collaboratively to deal with this epidemic. And so I don't think, and I think, to be quite blunt, I think we have to have more science in this country. The last, decade has been terrible for investments in science, and I think science is the only thing that can get us out of this. So India needs to think, ahead, and think about investing in science.
Great. So as Ashok said, we have tons and tons and tons of questions more, but we are we have run out of time. So I'd just like to start by thanking all of you for being here and taking your time to answer many, many questions on a very wide range of topics. And so Professor Gautam Menon, Dr. Dhananjaya Dendukuri, and Professor Satyajit Mayor, thank you very, very much for this session. I just want to summarize what we've discussed through this evening. The first one is that there is a lot of information that is now available because we've had a year of this whole nightmare. We understand much more about the mechanisms of how the virus works, and therefore it is possible to target the processes a lot better in all aspects of managing the disease. And there is a lot of work happening in that space as well. I think it was one of the things that came up very clearly is there is only one thing that will help us really prevent, and that is masks, triple layer, or maybe in a few days we're going to move to the 4-layer, but nevertheless, that seems to be the one thing that is completely in our hands. We had a very fascinating look into the variants of this virus that have evolved, and I think that gave us a good understanding of a number of definitions. And thank you, Gautam, for clarifying those. At least it now starts to, when we read an item in a newspaper or listen to someone on the TV, at least we will understand what is being discussed. As far as testing goes, RT-PCR would continue to be the gold standard, but there is a case for rapid testing. And then the choice of when do you do rapid versus when do you do, and when is it necessary to do a more detailed test, all of which is going to be a dynamic decision that needs to be taken as we're going along. The need for a lot more labs and the risk we stand in the propagation of this disease across the country if we don't build up testing capability across all parts of the country. We looked at genome sequencing and its necessity for us to be able to stay ahead of this virus if we can. And it was very heartening to note that NCBS is at the heart of this consortium of labs who are coming together to increase our ability to do the genome sequencing. Of course, it does involve also the build-up of a lot of technical capability, but we'll get there. We looked at vaccines, and it's really interesting that within a year we've actually got 6 different types of vaccine that have come out. And again, all of this would not have been possible if we did not have experience behind us over the many last years. And that knowledge is available globally to everyone. We came back again and looked at how did this whole effect and how we as a human species have exploited the earth and its resources much more than we should ever have done. And therefore, it's our responsibility now to do something to try and set right this balance as much as we can. And finally, a sense of hope, a ray of hope as global collaboration continues to work on dealing with this whole thing, and also to start thinking about what if, as is inevitable, there is another set of these pandemics that steps in. We can only assume that it's going to be worse, but is there some way that all of us put together can deal with it? And lastly, I think a message for all of us, which came across clearly from everybody: self-help is the best help, and prevention is the best way and the only way forward. So being aware, making sure we test when we need to, taking the vaccination, understanding that there are risks still. It's just because we've got the jab does not mean that we are immune yet. And there is a lot of work on medicines and so on, but it is still, the most important still is that we mask ourselves and make sure that A, we don't get infected, and B, we do not infect others because we could be asymptomatic and still be carriers. So we don't want any of that to happen. With this, once again, thank you all, and thanks to our audience for staying with us. Thank you all for being part of this very interesting session.
Thank you.
Thank you. Bye-bye.
Bye.